is this is hopefully going to be a little bit interactive. So we have the chat forum open, and if I'm going through the presentation and you have a question, please put it to the chat forum so we don't get a lot of background noise. But please feel free to ask questions as I'm going through, and myself or Blaze, who's running this webinar, will endeavour our best to answer those questions. Okay, so I'll start with our new test for pre-implantation genetic testing for aneuploidy, PGT-A. So PGT-A, it's the world's first algorithm specifically using artificial intelligence for aneuploidy screening. And ERPeak is endometrial receptivity testing. Both these products will be formally launched at Fertility 2019 in Birmingham. So hopefully the people attending will be able to come along to that and ask us more questions. But we'll go through some of these new tests right now. So what is it? What is PGT-A? So we've had a whole spectrum of developments in PGT-A, or PGS as it used to be called over the years, from the very first fish cases to the very first cases where we were able to look at all chromosomes back in the early 2000s, to the first case where we were able to look really sensitively at all chromosomes using a technique called array CGH, and that was a case that was run in the UK back in 2008. And then the technology moved on in 2014, 2015. We're able to use what's known as next generation sequencing to look for chromosome counts in embryos. So next generation sequencing. So it's a kind of buzzword about what we do, and what we're not doing, to be absolutely clear, is sequencing embryos. We're not sequencing every single base pair of an embryo, and we're not doing full in-depth know what every single base pair will be for the embryos in the future. And this is certainly going to be something that we're able to discuss in future in further depth. So we have our genome here, which we cut up into fragments, and then the DNA of each fragment is sequenced with each individual base pair, and then we align that along to our genome. What we don't do when we align this to the genome is we don't sequence every base pair, we just count chromosomes. And this is the way that the technology works, and it's really robust and it's been really reliable. So we generate our data on these next generation sequences, and then the DNA is converted into an image. And this is the way we've been doing it for the last few years. And it's good for us humans because us humans are really good at pattern recognition. We're really good at looking at something, maybe like when you look at an embryo and look at an embryo and see how the embryo is, different grades. We can train ourselves to that, but we're not really good at doing it reliably and robustly. So we create our images, and then we have to call to see whether something is normal or abnormal. And this takes some subjectivity and some human element of the analysis, and then we write reports. Maybe what I'll do is go through this and show you some of this actually in practice to see how this works. So this is what a result looks like for the next generation sequencing. And if you go along, it shows you each chromosome, chromosome 1, 2, 3, and a green line which shows you gains of chromosomes, and a red line which shows you loss of chromosomes. And that's how we've been doing this, and you get these nice pretty patterns, and up here you get a clear image which is a 46XX, so we know that embryo is female. Now in the UK you don't actually get the XX, you just get 46, so we don't tell you it's female because we can't, because that's gender selection, but we do develop this and tell you that that's our result. And at the bottom down here, there's another nice pretty image. There's no gains, no losses, and that's a 46XY. So fairly straightforward. So the next one I'll show you. Now, does that look like the previous ones? Well, it does look like the previous ones. It's a nice straightforward pattern along here, and then there's a change here. So instead of having 2 copies of each chromosome here, you have 1 chromosome X and 1 chromosome Y. So it looks like a male. But actually it doesn't look like a male because it's got a loss of chromosome 22 here as well. I don't know whether you can see that here. So it's a bit small, but actually there's also a loss of chromosome 22. And that's the sort of thing that we need to do double checking and then a further checking whenever we run a case, because you have to have multiple checks to make sure that you're not missing simple errors like that. And the software does help us. So you can see here we have a loss of chromosome 22. The software is good at making changes where you get full changes of all chromosomes, and it's good for pattern recognition. But the software that we've been using up until now doesn't do some things that are clearly there. So you can see here that this is a chromosome 1, and there's a partial loss of the bottom of the Q arm of chromosome 1. Now, this is a mosaic change, so it's not present in all the cells of the embryos, and there's part of a gain and part of a loss. And the software clearly has missed this. And this was because the software started off being very conservative and not being able to call really subtle changes. And again, here you have a change where you have a gain of part of the end of chromosome 2 followed by a loss. And once again, the software has not been able to call that. And it's a kind of combination of the existing algorithms we had when this software was developed 5 years ago, and a combination of the manufacturers of the software being very conservative in what they'd allow it to call and not, so as to avoid liability. But it's not great because it then requires human subjectivity to go through and analyze these changes. Of course, everything that I've shown you before is the pretty data that you always see. But actually, quite often you get data that looks like this, and you see that there's maybe small deviations from the normal, so small deviations or gains or losses of parts of chromosomes, and as you go along, you might say, you know, well, there's a little bit below the line here, some above the line, some more above the line here, and it becomes really difficult for the human eye to make a subjective call on this. Again, here you can see this is less noisy data, but you know, is this something that is clinically relevant here? This loss of chromosome 6. Again, a subjective call here. So what we've done is go back to basics and start off with the actual data coming out of the sequencer rather than anything else. So we're trying to remove the subjectivity, standardize what we do, reduce risks, because you know humans do make errors occasionally, and you need very complex systems to avoid those errors. But mainly what we're trying to do is reduce the noise, so we know what the noise is and improves the signals. And whenever you're doing these tests, it's all about increasing the signal to noise ratio. Also, just taking a different attitude to how we start this off and establish what we call truth. So this is the way that PGTai works. So the data comes out of the sequencer, and instead of going into a program which shows pretty graphs, it doesn't. It actually goes into a mathematical model, and we don't actually often look at the profiles now because we're just relying on the mathematical model and the statistical value to see whether something is called. So we started off with a completely different rationale. So Illumina and the other people that make these softwares started off with very small data sets, maybe 100 cells or something like that, to see what is their truth of gain and loss. We didn't do that. We started off with 1,000 embryos that had resulted in a phenotypically normal live birth, and that was our truth set. And then we took translocation cases, so they're cases where we know that there's a gain or loss, or cases where we know there was known abnormality, aneuploidy. And we started off with about 2,300 truth embryos, and then we've tested it with large numbers of data sets. Like some poor guys in California had to look at 10,000 samples to confirm this. And this uses artificial intelligence, and it's a bit of a buzzword, but there's a bunch of things that we do. So we use artificial intelligence, and it relies on computer learning to make decisions, and it's used all the time in various things in our life. And what we've done here is taken our big data set, and our first data set had 5 billion data points, and that is our truth set, and the computer programmers learn from that truth set, and then you can keep on iterating it, add in more data, and improve the quality of your results. It also becomes completely automated, so there's no need for human intervention, and the software as it is now will provide a complete carrier type straight from the analysis. It's already been used actually all the time in your life. You're probably using it most days. I mean, some of the very straightforward things that you see that are out there, Siri, or if you've got an iPhone, you press it, it uses artificial intelligence to work out from the noise and the words what you're actually saying to it, and it's pretty good. It's pretty good. Alexa is just the same. You come in, it learns from your voice what you're asking for. So when things are correct, it comes back and asks and doesn't need to ask them again, and it learns from that. And that's what we're trying to apply here for the first time for PGT-A. So we're leveraging our big data. Like I said, we started off with a data set that was a couple of billion data points. That's not actually big data in some other applications, but it's still pretty huge. But the thing that we're doing now, instead of eyeballing looking at these profiles, it's statistics, so it's p-values rather than a human analysis. Also, because it's automated, you don't get the transcription errors where somebody's written 45 when it should be 46, etc. And we've used our truth data set, which is our live birth data set, which is completely different from people have been done before. So this is our white box. This is our truth data set. This is our 1,000 live births, 650 known aneuploid embryos. These are embryos that had ended up in a miscarriage, 250 translocations, and 250 places where we'd mix DNA to create artificial mosaics, which is about 2,300 samples to start off with. To give you some idea, the previous softwares have started off with between 10 and 100 samples to start off their truth set. So trying to show it how it works. So you have to build a data set. So you start off with one euploid embryo and it's a bit noisy, and then you start adding in more. This has a different sort of noise, and we start to learn where the noise is. And after a while, you end up with a real large data set to show where your noise is and where your actual signals are. And then you can start to work out the stats of this. So you can see it's a general bell curve. The most of the data is along the 2 copies, and then it decreases. Then once you have that statistical model, you can apply that to changes. So this is our model of a sample. Now, this might be a clear gain that you could see, but you know, is this real here to the left as a change? So once you put your very large data set of normals in there, you can start to see that this is a real change and this isn't a real change. It could also call sex chromosome aneuploidies, which are really difficult. So we can find monosomy X, we can find XXY, triploid, as well as XYY. So it doesn't all just do your autosomes, chromosomes 1 to 22, but it will do all chromosomes as well. So this is an example of some noisy data, and this is what I showed you before. Now we use the PGT-A algorithm and we see how that applies. So instead of being human and subjective and spending a bunch of time and having a bunch of different people try to make an opinion on this, we're just using mathematical modeling. And actually what happens is some people are optimists, some people are pessimists. The pessimists will say, well, that's completely abnormal. And the optimists will say, yeah, you know what, I think that might work. And actually, that's one of the problems you have with human analysis. So now we have, this is our PGT-AI, and it will tell us that this is a true mosaic loss here, a true mosaic gain, and another mosaic gain. And it actually comes up and tells our karyotype. So our karyotype is 46XX with a mosaic loss of 1, a mosaic gain of 3, and then a mosaic duplication on chromosome 11 and a statistical value. Don't worry, this is not what your reports will look like. They will just say 46XY, mosaic 1, mosaic 3, mosaic 11. So your reports will look exactly the same, but you can see there will be an awful lot of modeling going in behind this. Another one, this is the one I showed you before as well. This is chromosome 6. Now, was this loss of chromosome 6 significant? Well, let's see what PGTai says. Well, it goes through and it does look at it and it says, yeah, this is significantly different from what we've done before. And now it has a karyotype of XY with a mosaic -6. And we can see that it actually won't come out and say XY because it's in the UK, so it'll just say 45. Again, another noisy sample. This time the noise is all within what we expect of noise to be. So you can see this is a normal sample, and that's where the software is really going to improve things, where things that we might have struggled to call normal, it's much more confident now because we have a really good mathematical model. So per 1,000 biopsies, the specificity is improved. So these are specificity, this is what you've called abnormal or mosaic, and we can change to euploid now. And we've taken our truth data set of euploids and just run it on that truth data set. So we're going to have an improvement in the number of euploids. Yet again, at sensitivity, things we might have called euploid, we now can be more certain that they are mosaic or aneuploid. So we've ended up with almost a 10% improvement in accuracy. So hopefully that will show clinical benefit at some point in the future. So PGT-AI, so what is it? Well, it uses artificial intelligence and big data, machine learning to create algorithms to take away human analysis of PGT-A and uses just now statistical mathematical modeling. It's in small data sets and used pretty looking charts to let the lab call the changes. It now has a really large 2,300 biopsy truth set, and it uses the statistical reports and produces a full karyotype. It improves the accuracy of what we've done, and we hope that will lead to a higher live birth rate per patient. Although obviously, because we've just introduced it now, that's hard to say for certain. The other thing which is really great is machine learning. It's not something that's fixed. Previously, you would write an algorithm to produce your nice pretty charts for PGT-A, whereas this, you can keep on adding. If you can keep on getting more data from known truth. So embryos that resulted in a healthy, phenotypically healthy live birth, embryos that resulted in a miscarriage, and you can apply them into the model and keep improving how the model is able to work. That won't happen all the time. It will happen in iterations where we get to a next release and then we move forward. So I'm just going to move on a little bit to talk about endometrial receptivity testing. So completely different area now, and we're moving away from embryo aneuploidy to talk about window of implantation and the endometrial, optimizing the endometrial transfer. So the endometrium, as it goes through the menstrual cycle, changes, and there is a particular period of the menstrual cycle called the window of implantation, when the endometrial is in the perfect time for implantation, and this is usually 8 to 10 days after the menstrual cycle. But you know, as humans, as women, everybody is different, and some people will have a window of implantation that occurs earlier, and some people will have a window of implantation that occurs later. And in approximately 30% of women, the window of implantation changes. So if you're implanting an embryo and somebody has a later window of implantation, you might end up with an unsuccessful implantation because you've just got the timing wrong. And there is some evidence to suggest that you get increased pregnancy rates if you can time the window of implantation for the correct embryo transfer. So, which patients do we use it for? So, recurrent implantation failure patients are the best patients for this particular test. So if you've had a patient and you've got, say, a really good, morphologically good grade embryo, or even better, a morphologically good grade euploid embryo, then this is a good test if that embryo or previous cycles have failed to implant. So you're doing everything right. You've got a good grade euploid embryo, but it's failed to implant. It might be the time to look at the endometrial receptivity to work out whether you need to shift that implantation backwards or forwards. And there are several publications and retrospective trials demonstrating the clinical efficacy of repeat implant of endometrial receptivity testing. The other one, which is an interesting one, is economical model. So if you have one euploid embryo, that's a very, very, very valuable, both kind of emotionally and biologically and physically, very, very valuable resource. And you want to have the best opportunity to get that euploid embryo to transfer. So putting a euploid embryo in with a endometrial receptivity test can really hopefully improve the chances of getting a pregnancy. So this is a new genomic test we've introduced, and what we've done is we've gone into the literature, and we're working with people who've been in this field from the very start, gone into the literature and looked at all the genes that are associated with endometrial receptivity and endometrial growth. And we have a test of 48 genes that we can use to differentiate between the different stages of implantation. And it measures things like expression of hormone-regulated genes, and it's on our custom developed and validated platform. So how does it work? It's pretty damn simple. So you do an endometrial biopsy as per normal. We have a kit that we send out to people, very simple, a little bit of liquid in there. You just stick it in the freezer. It doesn't have to be shipped on dry ice or cold or anything. You can just go room temperature. It goes to the laboratory. We run our test, issue the report, and then you can make a decision of whether you need to change the implantation timing for subsequent cycles. So each kit just contains the sample collection tube, a biohazard bag, our test order form, sample preparation and shipping instructions. And then you end up with a report that looks like this. So we've tried to make it as simple as possible. So you have 5 outcomes that you can have on the report. So this is the test result. This is if the test is what we call pre-receptive. So that's if the window of implantation is shifted to early, then this is our normal receptive state. So, and this is post-receptive. So if you get these different results, it could be that this sample, you've taken it at our normal time of implantation, but actually this lady was pre-receptive. So next time you'd have to shift the window of implantation to longer. This one is receptive at our normal window of implantation. So this really wasn't the cause of the repeat implantation failure. This one is post-receptive when we ran the test. So it's too late for implantation in this lady if you use the normal window of implantation. So next time you run a cycle, you shift it to an earlier. And then this is non-receptive, which is more unusual, and it indicates that the window of implantation just isn't right for that particular lady. Then of course occasionally you can get no results. So it just gives you some fairly straightforward indications of what you might expect in the test results. So you end up with the test. We give you some information here and some guidelines as to how it works, and this is the test there itself. So it's been a very brief little webinar. We've given you some details of our tests in terms of endometrial receptivity, but also some information about our new test for PGT-A, PGT-AI. We will be moving forward and producing developments of our PGT-AI algorithm in 2019 and 2020. Also, endometrial receptivity, there are a number of developments that will be coming out, and we'll get back and talk to you folks about that as they come through. And finally, just to say that we'll be present in Fertility 2019 with the team if you want to talk to us. So with that, I'd say thank you very much for your time. Now, if anybody wants to ask any questions about what's gone on, please hit the chat forum and enter your message now. Thanks for the message, Natalie. If you have any other questions, you can send them to Blaise or myself. I know that I'll have to check with Blaise whether the presentation is being recorded or not. The only other thing, but frankly, I'm going to have to apologise. For some strange reason, I had some dogs barking in the background, so apologies for that. Okay, so some more questions. Okay, Natalie's question first. Do you expect the timing of receptivity will remain the same between cycles? So as far as I know, the evidence is that if you have an earlier window of implantation, it remains earlier. That's just the nature of the physiology of the women. And that's why the test works, because if it changed between cycles, say you did your first test and it had a post-receptive, so you moved the second one to an earlier time. If it was varying between cycles, that wouldn't allow the test to work. So Xavier, thank you. Will you expect the time to report to be shorter with PGT-AI? We certainly hope it will, because we have. So when we get those pretty looking charts in a PGT-A test, you have to have 2 analysts independently read those. You have to have them checked off by a 3rd analyst, and then you have to have the lab manager or lab director sign off on that. So you have effectively 4 people checking each of those reports, whereas now we'll be able to upload these to the algorithm and get a result in a few minutes. Hopefully that will enable us to get back to you guys a bit quicker, because we know turnaround time is really important and really important to your patients. Any other questions? Okay, if there's not any other questions, then we'll finish the webinar now, and just really thank you for attending, and hopefully I'll see you at Fertility 2019, and if you've got any more questions at that point, then I'll be more than happy to help, or if you've got any questions, you can contact myself, Blaze, or your local CooperSurgical representative. All right, thank you for your time this morning.